Prior to beginning the activity, please be sure to review the faculty information and disclosure statements, as well as the learning objectives. After listening to the activity, complete the post-test by clicking the Earn Credit link in the episode description. Downloadable slides and resources are also available. The following presentation is copyrighted by Medscape. No use, broadcast, or recording of this presentation or any part thereof is permitted without the written authorization of Medscape.
The following presentation is part of a certified educational activity provided by Medscape Education and supported by an independent educational grant from Arginex.
This program is presented by Medscape Education Global.
Hello, I'm Isabel Pena. I'm a rheumatologist and assistant professor at the Ghent University in Ghent, Belgium. So welcome to the program. It's titled IgG-Mediated Autoimmune Disorders, a clinical update on novel therapies targeting FcRn. So joining me today in this discussion is Professor Rohit Agarwal. He is Medical Director and Professor of Medicine at the University of Pittsburgh in the United States. Welcome, Rohit.
Thank you.
Why it's important to cover this topic today. We all know autoimmune diseases affect about 10% of the general population. We know the pathogenesis of autoimmune diseases is driven by autoantibodies targeting self-antigens and leading to tissue damage. Autoantibodies can be of any isotype. We know that IgG-mediated autoimmune diseases are mostly driven by IgG, as I said already, and that IgG has strong effector functions. So I think we need specific and rapid therapies that can eliminate or reduce at least pathogenic IgG autoantibodies. So Rohit, can you explain me briefly why IgG-mediated autoimmune diseases, what exactly is the role of the neonatal Fc receptor?
Yes, so neonatal Fc receptor, which we call as FcRn, basically these are widely expressed receptors on endothelial cells, on hematopoietic cells, immune cells. The key function is to recycle the immunoglobulin, specifically IgG. And if you block that FcRn, what happens is this recycling of immunoglobulins stops and these immunoglobulins are degraded. So the levels of IgG goes down. But we are also interested with IgG is the levels of pathogenic immunoglobulins, which is what our diseases come from. So when you drop the immunoglobulin levels, the pathogenic antibodies goes down, and that hopefully helps our patients.
So can you tell me what's the rationale or the function of IgG autoantibodies in the story of myositis?
Yeah, so let me first, for our audience, tell what myositis is. So myositis is a rare autoimmune disease with a primary target of muscle, but it also targets other organs such as skin and other entities. What we've learned in myositis is that we have these myositis-specific antibodies, which appears to be pathogenic in nature. So what we have seen is when we reduce the level of antibodies, the disease gets better. So there's a significant role of these immunoglobulins or antibodies in myositis. We hope that if we specific targeting of these immunoglobulins, we can actually improve our patient's life by decreasing these pathogenic antibodies.
So it makes sense to target FcRn.
So, Isabel, can you tell us what Sjögren is and what's the role of FcRn receptors or immunoglobulins in Sjögren's?
So Sjögren's is a progressive autoimmune disease. It's characterized by lymphocytic infiltration of the endocrine glands and immune-mediated dysfunction of the endocrine glands. And if you look into the salivary gland, then we see that innate and adaptive immunity is both involved. And this leads to production of plasma cells. They produce autoantibodies, and those autoantibodies form immune complex, further fueling the vicious circle of immune-mediated inflammation. So it seems logical by targeting FcRn, decreasing the autoantibodies, that this can tackle the immune-mediated process. So, Rohit, what are the most recent efficacy and safety data of FcRn blockade in myositis?
So there are two major clinical trials that is going on, and these are both of them are phase 2 studies. So one is arfgatigomad study, which is Alkivia study, and another one is nipocalimab, which is Pyrevia study. And fortunately, this EULAR, we have results presented on Alkivia study, so I would like to present those results to you. So first of all, the design of Alkivia study. It's a very standard phase 2 clinical trial where you have a randomized patient 1:1 to weekly subcutaneous efgartigimod versus placebo on the top of standard of care immune suppression. So these patients, most of them are on immune suppressive drugs, to begin with. These include 3 different types of IAM, which is idiopathic inflammatory myopathies, is dermatomyositis, antisynthetase syndrome, and IMNM, which is immune-mediated necrotizing myopathy. And the primary endpoint of this trial is a 24-week, which is total improvement score called 2016 ACR-ULR total improvement score called TIS. And the way this total improvement score works, it has 6 components, as you know, in Sjögren's, similar to Sjögren's, and which include manual muscle testing, extramuscular global, physician global, patient global, muscle enzyme, and health assessment questionnaire. And you combine them to get a total improvement score. And we enrolled about 89 patients in this trial. So what were the key results? The key results were that in the treatment group, we saw a mean total improvement score of 50, which is quite a lot, as compared to 36, around 35, 36 in placebo group, with a highly statistically significant results. And what's really interesting is that the graph separates out very quickly, as early as 4 to 8 weeks, graph separates and continue to separate. So we're really excited seeing these efficacy results. Remember, this is still phase 2 study. Now, what about the safety signal? That's really good news as well. There was no new safety concern as compared to what we have seen in previously in FcRn receptor inhibitors, as you know, are FDA approved for other treatments. So it was really well done study with no major safety signal and very well tolerated by the patient. It's a sub-Q injection, so there was no problems that we have seen in this trial so far. The second trial with nepakulimab, which is Spirivia study, essentially this is also very similar phase 2 design with 1:1 randomization, but here the drug is IV with infusion every 2 weeks. And there are also patients who are on background immune suppression. So what you're giving them is IV nepakulimab every 2 weeks or placebo infusions every 2 weeks for the period of time. And the other thing is the total improvement score, which is the same outcome used with Alkivia study, is the same outcome used here. The key difference is there they were using mean total improvement score between the treatment and placebo. Here, this is a threshold response that those patients, a proportion of patients who improve by total improvement score of 20 or more, which is the minimal improvement by the time point of the study. So that's the key difference. But unfortunately, I don't have the results of the Spirivia study to discuss. So for that, we would have to wait. But we're hoping that study will be completed soon and we'll have results to discuss with our audience soon. Can you tell us about the clinical trials going on about targeting FcRn receptors in Sjögren's?
So we have 2 trials which evaluated FcRn blockade in Sjögren's disease. The first one I like to mention is the ROW trial. It's a phase 2 placebo-controlled randomized trial with efgartigimod, in which they included 34 Sjögren's disease patients. It was a 2-to-1 randomized trial, and they treated the patient IV weekly. There were 23 patients in the treatment group and 11 patients in the placebo group, and they were treated for 24 weeks. So the primary endpoint was the CREST response. It's a quite novel composite score. It has 5 items: systemic disease activity, PIROs, T-gland function, salivary gland function, also IgG and rheumatoid factor. If we look at the primary endpoint, then we see that in the treatment group, 44% response on CREST. If we look in the placebo group, it's only 11%. So this gives us a treatment difference of at least 34 percentage points, which is quite good. If we look at the individual items of the CREST, then we see that in at least 4 of the 5 items, there is numerically higher response in the efgartigimod group. So this is good news. Then if we look at all the secondary endpoints like change in SDI, clean SDI, the CSTOL, which is comparable to the CREST, but it's a bit different. It puts more weight in the systemic disease activity and in the PIROs. All these secondary endpoints are also numerically higher in the efgartigimod group. Then if we look at the safety data, then we can say that efgartigimod is generally well tolerated and that no new safety concerns were signaled. And then we have the other study with nipocalimab. It's called the DAHLIA study. It's also a phase 2 trial. It's the largest study. There we have 3 groups. We have a group with placebo, a group with 5 milligram per kilogram, and a group with 50 milligram per kilogram. And in each group, more or less 50 participants. And then they received IV nipocalimab every 2 weeks through week 22. The patients had moderate to severe Sjögren's disease. They had also anti-SSA antibodies, and their primary endpoint was change from baseline in ClinSI at week 24 and also safety at week 30. So if we look at those results, then we see that they saw a least square difference of minus 2.65 between the placebo and the highest treatment group. They had also secondary endpoints which were reached. And if we look at the safety concerns, we can say that the treatment was well tolerated and that there were no major safety concerns, as was for efgartigimod. So these are comparable.
What are the implications of these clinical data in Sjögren's?
I think that both studies suggest the benefit of IgG reduction in Sjögren's disease, and it highlights, of course, the potential clinical impact of FcRn inhibition. For both products, both nipocalimab and efgartigimod, they're now running phase 3 trials. And I have to say that for efgartigimod, the first trial, the phase 2 trial, was with IV weekly, which is quite a burden for patients. But now the phase 3 trial, which is called the Unity trial, is with subcutaneous administration. So it's much easier, and it's for 48 weeks. And then we also have the Daffodil trial, which is with nipocalimab. And for you, what do you think the implications will be for myositis with FcRn inhibition?
Yeah, so I think myositis implications is as huge as in Sjögren's. As you see, myositis is also a disease of unmet need. We don't have any FDA-approved treatment for all indications. We have one FDA-approved treatment for dermatomyositis with proven results in randomized controlled trial. So these two trials are very exciting for us, specifically with the Aclivia study showing significantly positive results without major any safety concerns here. But the real test is phase 3. So we're really going to see the results of the phase 3 of hopefully both the studies going forward to phase 3 in future. But it really provides a hope that by blocking FcRn receptor, we increase the degradation of immunoglobulin, including pathogenic antibody, resulting in decrease in the levels of pathogenic antibody that in turn will hopefully improve the patient's clinical symptoms like muscle weakness and skin rashes. So we're really hoping these, hope at least is that we'll have our patients have some treatment in future.
I had the impression that for both Sjögren's and myositis, it's a bit the same story. No approved therapies, autoantibodies, reducing antibodies, and probably have some effect on the patient's symptom burden. No?
Yep, absolutely.
So thank you, Rohit, for this great discussion. I would also like to thank you for participating in this activity. So please continue on to answer the questions that follow and complete the evaluation. Thank you very much.
This program was presented by Medscape Education Global.